I'm Janita Banasundara, research scientist working on plant proteins from Agriculture and Agri-Food Canada, and the moderator for today's webinar. Uh, Title of the webinar today is Defining the Quality of Plant-Based Proteins, Challenges and Opportunities for Pulses, with the learning objectives of to understand how protein quality is measured and how it influences allowable protein content claim on foods, and to provide an overview of the factors influencing protein quality. To provide you a brief introduction of the webinar today, consumers are demanding foods with higher contents of quality protein, particularly plant-based proteins. Additionally, national dietary guidelines are emphasizing the importance of plant-based proteins on human diet as part of an overall strategy to encourage dietary patterns deemed to be more conducive to optimal human health. These patterns can include protein-rich, whole foods, as well as processed foods low in saturated fat, sugar, and sodium. In Canada and the United States, positioning foods to consumers as being rich sources of protein requires substantiation of the quality of the protein in question. Protein quality in general is a function of how well the amino acid pattern of the food matches human amino acid needs and the extent to which amino acids are digested, absorbed, and ultimately made available to meet the needs of the consumer. Today's webinar will provide an overview of the current and proposed methods positioned to measure protein quality and summarize the impact of various parameters on protein quality, including processing, blending, varietal selection, agronomics factors, and other things related to consumers such as age. Let me introduce the webinar presenter today, Dr. James D. House. Dr. House is the professor and head of the Department of Food and Human Nutritional Sciences, Faculty of Agricultural and Food Sciences, University of Manitoba, Winnipeg, Manitoba, Canada. He has authored and co-authored 275 publications, book chapters, and conference abstracts and proceedings in areas related to nutrition and metabolism, including those linked to the study of factors influencing plant protein quality. A few housekeeping issues before the presentation begins. Participants will be muted remotely during the presentation. However, the chat tool of the right side of your screen is open throughout the presentation to send any questions if you have. Dr. House will answer your questions during the designated Q&A time at the end of the presentation. So without further delay, I invite Dr. James House to deliver his presentation.
Great, thank you, Janita, and hopefully everybody can hear me okay. If not, please just let me know via the chat function. But I just want to take this opportunity to thank Janita for the wonderful introduction, and also to thank you for attending this webinar, and finally to AOCS for making this opportunity possible. So as Janita mentioned, the title of my talk will be Defining the Quality of Plant-Based Proteins, Challenges and Opportunities for Pulses. In terms of an overview, I'll be looking at providing a brief overview of protein consumption trends and the ways in which we position protein to consumers, and then specifically looking at the importance of quality. Then ultimately I'll go into a discussion of pulse proteins specifically, looking at some of the challenges and opportunities. And again, this is meant to be an overview of some of the issues that I feel are impacting the sector as we move forward. First of all, to begin with, why are we seeing such an increased focus on protein? I'm seeing that protein is being used as an umbrella term for a number of different items, but classically, protein is considered a nutrient. And when we think about it from a nutritional perspective, there's been increasing interest and increasing research to look at the role and importance of protein and the quality of that protein in promoting optimal performance, weight maintenance, optimal immune function, and healthy aging. And a lot of these factors intersect. And in particular, as we have an aging demographic within North America, these factors are resonating within consumers as we look at options for trying to enhance the healthfulness of the foods that we're consuming. And protein has a specific health halo around it, which is intriguing to consumers. We may not always understand why, but I think it's really important to recognize that as a trend, consumers are seeking foods with more protein. When it comes to looking at protein as a nutrient, we know that we have established requirement patterns. The current requirement patterns, if we were to position them, and I'm primarily talking about the adults' requirement patterns right now. So looking at folks under the age of 70, about 0.8 grams per kilo per day of protein is the requirement. And if we're older than 70 years of age, the current requirement has it at 0.8 grams per day, and that's the recognized patterns within the that Health Canada and the FDA have agreed upon. But there's increasing evidence in the literature that, in particular, as we age, the requirement in an aging population might actually be higher than that 0.8 grams per kilo per day. And we're certainly seeing significant evidence in the literature that is positioning a higher requirement pattern, in particular for aging. And that's in particular to start looking at means or ways of reducing what's called age-related sarcopenia or muscle loss, and a focus on higher quality and higher intakes of protein in an aging population has received greater interest as of late. We know how much we need according to current estimates, but how much are we actually consuming? This is an important question because if we start looking at dietary patterns and dietary intakes, and I'm just showing you data that was published in 2015, results from the N. Haynes studies within the United States. This is a national survey that is conducted within the US, and a review of the N. Haynes cycle between 2007 and 2010 basically estimated that the United States population on average is consuming approximately 40 grams of protein per 1,000 calories consumed. And so if your requirement is about 2,000 calories per day, you would be consuming roughly 80 grams of protein on average. What's interesting to note, though, of that distribution, approximately 70% of the protein is coming from animal-based sources, with 20% from dairy and approximately 50% from meat, eggs, and offal, liver, etc. Whereas 30% was coming from plant-based sources, including breads and rolls, pasta, and legumes. So 1.3% would be coming from legumes, including pulses. So of the plant-based sources of protein, a significant proportion in the North American diet of that plant-based source is actually coming from cereals, even though cereals are not necessarily considered traditionally as high protein sources globally, and cereals contribute a significant component of the total protein intake. So it is an important consideration. If we look at, that was the American data, the current analysis of the Canadian cycle, the Canadian equivalent to what's called the CCHS analysis is ongoing. These are some initial infographics that have been published by Statistics Canada about percent of Canadians who eat the sources of protein on any given day. And just to give you an idea of what are the primary protein sources, the top source are dairy products, with approximately 94% of Canadians who eat dairy sources of protein on any given day. Going down into meat at about 63%, eggs at 56%, and then finally getting down to legumes, of which pulses are included in that category, about 14% of Canadians who eat these sources of protein on any given day. So again, you'll see that it definitely is slanted more to a higher animal-based protein in the current Canadian and North American diet. Now, when we start looking at dietary guidelines, and so this is information that's meant to guide the way in which North Americans are consuming. In the US, we have the Dietary Guidelines for Americans, so the 2015 and 2020 cycle indicates that we should be consuming a variety of protein foods, including seafood, lean meats, and legumes, of which beans and peas, soy products, and nuts and seeds are contributing component of that. In Canada, we just released the Canada's Food Guide. This was just released in January of 2019. And the guiding principle within Canada's Food Guide was to eat plenty of vegetables and fruits, whole grain foods, and protein foods, and choose protein foods that come from plants more often. So this is a significant shift in Canada's Food Guide, which before this time had 5 different categories. Or 4 different categories. Essentially, it had milk and alternatives, meat and alternatives, fruit and veg, and grains. Now with the new Canada's Food Guide, moving away from the rainbow, we now have a plate, which those in the US would recognize a plate as being a part of the My Plate system. In Canada, it's a little bit different concept. Even though we're using the plate as the graphic, there are no serving sizes attached with this plate. Essentially, it's a proportional approach in that half of the plate is represented as being part of fruits and vegetables. And that's the recommendation, to have plenty of fruits and vegetables. Choose whole grain foods. Make water your drink of choice. And so you'll see a shift with an absence of the milk category in Canada's Food Guide. And now a section called protein foods, of which dairy is a part of that. And you'll see that there's dairy in the representation of this low-fat yogurt source. But there's a combination of animal and plant-based protein source in this protein food category. So when we look at, when we look back at other additional recommendations that are made in Canada's Food Guide, these include limiting highly processed foods. And if you choose these foods, eat them less often and in small amounts. Prepare meals and snacks using ingredients that have little or to no added sodium, sugars, or saturated fat. Choose healthier menu options, use food labels, etc. So this is meant to be a live document. It's a web-based resource that was released. But I think the biggest shift had to do with this introduction of this protein foods category. And I think that's the category where pulses are fitting right now. So when we look at plant proteins, what are some of the opportunities? Well, certainly there's an enhanced profile within the dietary guidelines, both in the US and in Canada. We definitely see an enhanced profile. In particular with the statement around having a protein category, and then a specific statement that we should be consuming plant proteins more often. So definitely a higher focus on plant protein consumption. We have regional advantages in plant protein productions within North America. Where I'm located in Winnipeg is in the start of the Prairie region. We have a competitive advantage in areas related to cereal grain production, oilseed production. So we have a significant amount of plant-based protein that's grown within Western Canada and also throughout the U.S. Great Plains area. So we have a significant amount of plant-based resources that is available for us to look at in terms of incorporating into the human diet. With those plant proteins comes a greater amount of variety. So we have functionality, we have enhanced nutritional quality attributes when we start looking at plant protein sources. So with that variety comes some unique attributes that can be tailored to meet specific consumer needs. And what has been increasing in focus with respect to dietary guidelines is this issue around environmental sustainability and greenhouse gas emissions. And there is the potential for better sustainability metrics. I want to make it clear that I'm saying potential. I think that there is a lot of data to support enhanced sustainability metrics, but there are some processes that we are engaged with in terms of protein production and protein positioning within the North American and global food supply, that we still need to be mindful of sustainability metrics, including water usage. But certainly nitrogen fixation and legumes, pulses as a legume crop fix nitrogen, and that can be a major contributor to greenhouse gas is the use of nitrogen fertilization if it's not used properly. So I think we have a significant number of opportunities when we focus around plant proteins. And some of this is meant to just give you a pictorial representation of some of the products that are available. It's not meant to be an endorsement by any means, but certainly you can see that we have a number of products that are coming onto the marketplace that focus around plant-based proteins, that are made entirely of plant-based proteins, including burgers, including beverages, pastas. These are all heavily, they're of growing interest by consumers. These companies are capturing new opportunities by positioning food products that consumers are demanding. And so I think that's one of the things that we need to be mindful of, is that there is tremendous opportunity within the plant proteins. But along with these opportunities, we have to be thinking of the whole picture. And so when I talked about sustainability of pulses, here's an example of some data that is providing evidence of a positive impact on the carbon footprint when you think about substituting pulses, in this case yellow pea flour, into traditional wheat-based foods. And so a pasta, a breakfast cereal, or a pan bread, if you think about those as traditionally wheat-based foods and you incorporate a legume in the form of yellow pea flour at different inclusion rates, and so 15% for pan bread, 53% replacement for breakfast cereal, or a 30% replacement for pasta, you actually get an improvement in the nutrition balance score. And so this is an approach that was used by the authors, Chowdhury et al., published in the journal Nutrients, where they looked at the density of the food and then divided that by the carbon footprint of that corresponding food. And using what they're considering as the nutrition carbon footprint score, they could show significant improvements in the scores. An increase basically shows that you have greater nutrient density with a lower carbon footprint. So that ratio, a higher ratio, is more beneficial. And certainly by including that yellow pea flour, we're seeing benefits there. This is just one metric of environmental impact and environmental sustainability, but it does highlight the potential for a good story to be told about plant-based proteins, in particular legumes like pulses. But they're not without challenges. And so when we think about plant proteins, we have some challenges. Then luckily for us, so for those researchers on the line that are engaged in research and looking at protein and protein quality and issues around plant proteins, challenges I view as opportunities, because when you start looking at challenges, we need to understand how to overcome these challenges. And some of the challenges include issues around variability. So looking at sources of variability within plant proteins, not only do we have variability in terms of composition, we have variability in terms of supply, variability in terms of price. So I think all of these are factors that we need to be mindful of when we're looking at plant proteins: variability in price and supply, quality, and functionality. We layer over top of that some of the regulatory challenges that are in existence when we look at how we can position plant-based proteins and ingredients to the consumer. And one of them that I want to talk about, is the focus of today's talk, is this issue around protein content claims, in which we require the use of a metric related to protein quality in order to substantiate these protein content claims. So when we talk about communicating protein messages, the first way we can do that is start looking at the nutrition facts panel. I'll just give you an example of a nutrition facts panel. This is an older nutrition facts panel from the US, and looking at kidney beans as an example. And so in this particular case, we have a serving size, we have a protein statement at the bottom that there's 8 grams of protein that constitutes 16% of the daily value in about a half a cup serving size. And so you can gain information from that nutrition facts panel. That it requires that the consumer be educated in how to use the nutrition facts panel, and there is a role for increased education to enhance our understanding how consumers relate to nutrition facts panels and, in fact, front of pack labeling information. But some of the other ways that we can convey information through the use of claims: origin claims, composition claims, symbols, and pertinent for today's talk is the issue around nutrient content claims, where we see, for example, a product with the statement that it's an excellent source of protein or it's high in protein. These are nutrient content claims, and as claims, according to both Canadian and US law, they require substantiation. And so how are those claims substantiated? Well, what evidence is needed to support nutrient content claims? It really depends on the jurisdiction, but essentially in North America, we use quality as the parameter to as the primary parameter to evaluate the source of the protein that's being used within a food to see if it can meet a protein content claim in conjunction with the quantity of the protein that's also present. So it's a combination of both quantity and quality that's important for substantiating nutrient content claims in North America. I'll explain more about that in just a second, but in certain jurisdictions, they only use quantity. So the total amount of protein, regardless of the source of that protein. So in the European Union, for example, that's primarily protein as a function of energy content that is a reflection of that's used to substantiate protein content claims. So in Canada, we use protein quality and quantity as the basis for protein content claims. And essentially, we use a protein rating system. So the official method that is required by Health Canada and is as surveyed by the Canadian Food Inspection Agency is the protein rating system, which is based on the protein efficiency ratio. And the protein efficiency ratio is essentially a growth assay that looks at the growth of a rodent model when it's consuming a specific protein source in comparison to the same amount of growth that a rodent, that a control group would be generating when it's consuming a high quality control protein source. In this case, they use the milk protein casein. So it's a very simple assay. You feed the protein over a 28-day period, and essentially what you're doing is you're measuring the amount of growth that that protein can support over that 28 days. And the higher rate of growth is reflective of a higher quality protein. In the United States, again, it's protein quality and quantity, and the primary quality component that's used is different than what we use in Canada, and so it's what's called the PDCAS, or the Protein Digestibility Corrected Amino Acid Score. And essentially what this method does is it looks at the composition of the protein from its amino acids. So you measure the amino acid content of the protein, and you relate those amino acids to a reference pattern. And so the better if your source protein matches more closely with the reference protein, in terms of a ratio, you would end up getting a value of 1 or greater, would indicate a score value that shows that there's no limitations in the amino acids present within that particular protein. Anything less than 1 would indicate that there's some limitations and would have a lower amino acid score. Once you have that score, you actually correct it by the protein digestibility, which is also determined during an animal model using what's called true fecal protein digestibility, essentially measuring the amount of nitrogen that goes in and the nitrogen that comes out of a rodent during a feeding assay using rodents. In the European Union currently, as I mentioned, it's an expression of protein content relative to energy content. Now, there is a proposed method that has been positioned by the FAO, and it's called the DIAS method. I know there's been a lot of interest in this method. It's the Digestible Indispensable Amino Acid Score method. Essentially, it's a method very similar in concept to the PDCAS, but instead of using the fecal protein digestibility in a rodent model, they use what's called the ileal digestibility coefficient, which is measuring how much of the amino acid that is fed is actually absorbed by the time it reaches the end of the small intestine, which is the primary site of absorption in animals. And then they use those coefficients to determine the actual score for individual amino acids. And so we're actually viewing the individual amino acids as the nutrient in question. And so you develop a limiting score based on the limiting indispensable amino acid that's available in that particular method. This method is currently not being used anywhere globally. It's proposed right now. There are groups establishing data to determine how well this method can be used. That data will then need to be modeled and placed upon the existing food supply within North America to see how feasible it is to move to this particular DIAS methodology. The challenge that we have in all of these methods is that they're animal-based models, and that's a major issue for some companies who are hoping to present quality protein sources in new food supplies that that don't require the use of animal testing. And I think that's a major limiting factor for the current methods that we use to look at protein quality. So as I mentioned, protein quality is a function of the amino acid composition and digestibility. So composition is how well does the amino acid pattern match human amino acid needs. Digestibility is to what extent are the amino acids digested, absorbed, and ultimately made available for metabolic demands. The PER, PDCAS, and DIAS methods all relate in some way to those two particular concepts. So an amino acid score less than 1 indicates that at least one amino acid is limiting. In pulses, typically the limiting amino acid is either tryptophan or the sulfur amino acids, whereas in cereals, they tend to be limiting in lysine. So classically, if you mixed pulses and cereals, they tend to offset in terms of their limitations, and you can actually increase the quality through a process of blending. So the classic beans and rice approach in terms of protein complementation, balancing the amino acid profiles. The other component beyond the amino acid pattern is the digestibility. So plant-based proteins typically present with lower digestibility because we have an encapsulating effect of cell wall, a presence of protease inhibitors, and a number of other factors that can influence digestibility. So really, when you're looking at what factors influence protein quality within a context of what I'm discussing here, we have to consider both components. What's happening to the amino acid composition, what's happening to the digestibility or availability of those amino acids. So let me just go through a quick snapshot of the pulse sector with that as a background. So we've been talking about pulses, but just to make sure that we're all on the same page, what we're talking about here are the dry beans, dry peas, chickpeas, and lentils. And so essentially the dried seeds of legume crops, non-oilseed legume crops. So these would not include dried soybeans, for example, as a pulse. From a Canadian perspective, global we produce approximately 8% of world production, but we only consume about 7% of what is produced. So we are an export-focused economy. Global pulse production is about 82 million tons, and I think we have a significant amount of pulse that's being consumed, but there's tremendous opportunity to look at pulses as a source of protein. But what I want to do is just focus the remaining parts of this talk on some of the factors that we know are impacting protein quality of pulses. And so when we talk about those two components, the amino acid composition and the digestibility pieces, each of those two components can be impacted by a variety of factors. So plant genetics, for example, can influence both the quality, the digestibility, as well as the amino acid composition. Agronomic factors can influence both as well. Processing and blending. So I'll give a couple of examples drawn from experiences within our lab and from others where they've looked at the factors that are influencing the protein quality of pulses. And again, this is meant to be primarily an overview, and we can certainly go into more depth during the discussion period. So when we look at genotype by environmental factors, this is just some data from yellow peas that we've conducted within Manitoba from 67 varieties of yellow peas that were grown in various locations within the same agronomic year. And this is just to highlight the variability within the amino acid score values using, and in this particular case, it's tryptophan as a limiting amino acid. And what we can see here is that we have significant variability in the amino acid score. And so when you start looking at the single estimates of amino acid score, you need to reconcile against the sources of variability. In this particular case, I'm just showing one source of variability is the plots where the origins of the yellow peas. And so these are all from different locations within Manitoba. So we're seeing significant variability in just the amino acid composition. And likely that's a reflection of the protein content as well. So certainly in this particular case, we're seeing the relative frequency map of the protein content of the same peas goes from about 19% up to a high of almost 26%. And so considerable variation and variability in the protein content and therefore the amino acid composition of the proteins. one of the things that we're gaining more information on is whether or not those changes of the amino acids are happening as a function of the protein content, or are we seeing shifts in the relative amounts of different storage proteins? Because there are different forms of proteins within legume crops, and they differ. And so you can see perhaps potentially different shifts in patterns of storage proteins within legumes under different environmental or genotype conditions. And so that variability is important to understand because it can help feed back into breeding programs for pulses to guide the selection of legumes, for example, or peas or pulse or other forms of pulses like lentils that could then be positioned for specific usage by segments of the marketplace. So for example, if you're producing an isolate from a lentil and you want to be focused on a higher protein pea, by understanding the sources of variability and also the composition of that protein that's extractable, you can then, if we know where that variability is occurring, we can select for it and then perhaps focus in on those higher protein or higher amino acid lines that would be better suited for a specific endpoint that our consumers are looking for. Processing plays a number of roles in impacting quality, both positively and negatively. So certainly when we look at processing methods, we're talking about physical methods such as fractionation, drying, milling, extrusion. Certainly these will have an impact on the digestibility of the protein. For example, milling and extrusion can lead to physical changes, the disruption of the encapsulating effect of the cell wall on the proteins, and so it makes it easier to digest once it's consumed. Fractionation, if you're looking at physical separation such as air classification processes to create a protein concentrate from a pulse flour, you're removing fiber fractions that can have a suppressive effect on the overall digestibility of a particular protein source. So processing can have effect on digestibility. It can also lead to shifts in the amino acid pattern if you're causing a shift in storage form of that particular protein. If your method is selecting for a particular protein that's located within that legume or pulse. Biological processes, there's a fair bit of work happening and looking at effective fermentation and enzymatic hydrolyses in terms of understanding how they can impact on the quality of proteins. Same concepts apply here. Enzymatic hydrolysis and fermentation can lead to shifts in the factors that impact the digestibility of the protein, can impact on, depending on the nature of the fermentation or enzymes that are being used, can actually break down the cell wall and can actually improve digestibility. Chemical methods such as aqueous extraction or acid hydrolysis can play similar roles as well. We do need to be mindful that heating and the thermal processes that we add during the protein processing of pulse proteins do play a role in particular impacting the quality from a nutritional standpoint. Just to give you an example of some of the impacts that thermal processing can have on the protein quality of pulses, here's some work that was published in 2017. And these authors found that if you looked at a combination of legumes, so yellow pea, green pea, red lentil, chickpea, and black bean, that if you subjected them to these pulses to either soaking or cooking, and then you looked at the trypsin inhibitor activity. And trypsin inhibitor basically is that particular activity has a suppressive effect on one of the main enzymes that is used to break down proteins. And so if you have a high trypsin inhibitor activity in your pulse, it will lead to a suppression of the digestibility of that protein, and you'd want to overcome that. And so soaking doesn't appear to have a significant impact on the trypsin inhibitor activity of pulses, but as soon as we cook them, you can see that heat transferred to those pulses is sufficient to significantly bring down that trypsin inhibitor activity. effectively removing that as a suppressive effect on the overall digestibility of that protein. You would still need to test that in model systems to confirm it, but trypsin inhibitor activity does correlate very highly with digestibility. But one of the things that we do need to be mindful of is that when we do add heat, we have the ability to go too far. And so that's something that we're always having to consider, is that too much heat can actually cause a disruption of the composition of the protein, and in particular damage some of the key amino acids such as lysine. The amino acid group of lysine is reactive to and can react with reducing sugars to form Maillard reaction products, and those products are not necessarily digested and used as lysine. They may be absorbed, but they don't actually contribute lysine to the host, and so they can't actually take advantage of that lysine. So too much heat can actually cause thermal damage to protein sources. So, and this is what this graph is showing from work that was done in the early 90s. So it's a balancing act, finding the right amount of heat to suppress trypsin inhibitor activity, but not going as far to cause damage to some of the key amino acids. And so that's one of the areas of active research right now, is understanding the combination of physical, chemical, and biological methods that can be used to enhance the quality of the protein that we are interested in from these pulses. Extrusion is another method, and so we've done some work on extruding a number of different pulse flours. Here's just an example of some work with pinto bean flour, showing the amino acid score, AAS, the true protein digestibility, or percent TPD, and then if you multiply those two values together, you get the PD-Cas. And so 0.73 times 0.848 will give you 0.62, a PD-Cas value. One of the things that you'll notice is that in the native pinto bean flour, we can measure the amino acid score because it's just a chemical method. I didn't report the data here, but one of the things we can't measure is the true protein digestibility, because rats will not consume uncooked or raw pinto bean flour. And so if you're reliant on a method that requires a bioassay of an animal consuming it, this is an issue, because first of all, the animal will not consume uncooked pinto bean flour. As soon as we extrude it, however, we get a very high protein digestibility of about 85%, and coupled with the amino acid score of about 0.73, you end up with a PD-Cas value of about 0.62. And so the goal for pinto bean flour is, what then are there ways in which we can enhance amino acid score, enhance that protein digestibility? Certainly baking does not seem to do that. It has a suppressive effect on the protein digestibility when we baked it as a cracker, likely due to damage in the amino acids that are, or the overall digestibility of that particular protein. Finally, another way that we can influence the quality of pulse proteins is through blending. And so here's an example of the impact of adding pulse flour to wheat flour on overall PD-Cas values. And this is just a schematic representation of what happens when you include green lentil flour, yellow pea flour, or chickpea flour at various inclusion rates to the overall PD-Cas, or protein digestibility corrected amino acid score. And so if you're starting off at, say, 0.4 as the PD-Cas value for a 100% wheat flour, as soon as we add a pulse, we get an improvement in that PD-Cas score, because essentially the wheat is limiting in lysine, but the pulses have more than enough lysine in them to overcome that limitation that wheat has. So by adding the pulse, we're adding lysine and we're increasing the score. And roughly when we get between 30 and 40% inclusion rates is when we tend to reach the maxima. And that's where we are now at the point where any further additions of the pulse will not improve the amino acid score, because we're now starting to target towards the limitation of the pulse itself. And so when we start getting closer to 100% pulse inclusion, that's when we start going towards the PDCAS of the pulse itself. And they tend to be limiting in either the sulfur amino acids or the lysine. But there is a sweet spot that you can model by looking at inclusion rates to just determine whether or not a particular inclusion rate will generate a theoretical PDCAS value that would allow you to make a particular content claim. So some of the key considerations for blending, you want to find ideal partners. Allergenicity and clean labels are major issues right now. I think that's one of the benefits of looking at pulses right now is they're from a perspective of clean label and allergenicity, they're viewed in a very positive light and as a potential competition for soy proteins, which have that, which are listed as one of the higher allergenicity components. You want to be mindful of still functionality, sensory attributes, cost, and availability. These are all key considerations when you're coming up with ideal blends to position within food products. Some of the other regulatory issues include harmonization of protein content claims. And so that's something that we've published on and others have published on, is the fact that Canada and the US have different methods. And so if you're producing a product in the US and you want to have a protein content claim on it, you have to use two different regulatory approved methods in order to substantiate those claims. And so ideally, harmonization of protein content claims. And so I do think that both Health Canada and the USDA will be moving in that direction, or the FDA will be moving in that direction to help to harmonize. And likely it's Health Canada moving more towards the FDA position of using, at least in the interim, the PDCAS value. Standards of identity is a big issue, and so I know that plant milks are certainly something that has been in the news last year when I think it was the Minister of Agriculture in the United States indicated that almonds don't lactate, so how can you have almond milk? But certainly the standards of identity is an issue. And with just reading an interest in the current issue of food technology, that one of the trends that they were mentioning is to nix the alternatives and to focus on the positive attributes of plant proteins and pulse proteins by their own right. Stop thinking about them as substitutes and start focusing on solely letting them shine by their own merits. And so this may or may not be an issue going forward as more emphasis and the consumers are already identifying that the positive attributes of new plant-based products. One issue that is likely not going away is the nitrogen conversion factor. Currently, pulses use a value of 6.25 times the nitrogen factor to generate crude protein. I see an era of continuing challenge to this value of 6.25, and it will be something that because I think most evidence is showing that for a lot of the pulse proteins and plant-based proteins, the true number is down in the mid-5s for nitrogen conversion factors. Some plant-based proteins are required to use a different value than 6.25. Almonds, for example, a value of 5.18. Wheat has a specific factor that they use. But it's only a few of the commodities that have a value other than 6.25. And so I think this will be an area that is going to get increasing attention as we see more competition in the marketplace for proteins. One of the things that I think our lab has been focusing on is looking at in vitro techniques for assessing protein quality. And just to give you an example, I talked earlier about the challenges of using animal models to determine protein quality and making that a requirement or required analytical method to substantiate protein content claims. Well, protein is the only nutrient that requires the use of an animal model for content claims. And so it's obviously there is a concern about protein quality because you want to have some way of protecting against very poor quality nitrogen sources that are absent of a particular amino acid, for example. If you just use the nitrogen conversion factor and multiply by the crude nitrogen content to get a protein value without having some way of assessing how well they're contributing to the amino acid supply, it could be an issue. But there are likely other ways that we can approach. And certainly in vitro methods to measure digestibility and multiplying that by the amino acid score, which in itself is a chemical or an analytical method, can generate an alternative. And we've been finding really fairly good agreement between PDCAS determined by the official method and then the in vitro PDCAS determined using a method, an in vitro method for measuring protein digestibility. Some of the other remaining hurdles, protein ingredient supply chain, price, availability, consistency, consumer acceptability. These are all going to be important factors that as the supply chain matures and more products become available, I think we'll see that these tend to become more defined over time. I think we're still going to be seeing some challenges with respect to this image of processed foods and highly processed foods and in particular dietary guidelines. The Canada's food guide is basically positioning that we should avoid highly processed foods. The caveat or the defining statement that tends to get not necessarily as much focus as the fact that it's highly processed foods high in saturated fats, added sugars, and sodium. And so is there a role for the food sector to start positioning plant-based food proteins that are products that address these nutrients of concern? And I think that's where the balance will likely come as we move forward in addressing the role of processed foods and contributing to a healthy diet. With that, I think I'll finish up my formal part of the talk. Just acknowledge the research team here, and particularly the few items that were individual that were mentioned there. Collaborators, including collaborators in Saskatchewan, Alberta, and in the US. Funding partners, in particular Agriculture and Agri-Food Canada, because they did provide significant support through their Pulse Science Cluster 2, and also the Global Institute for Food Security based out of the University of Saskatchewan provided funding. I'd also like to thank AOCS and to make a shameless plug for a Pulse Forum that is being held this November in 2019, November 5th to 7th, the Pulse Science and Technology Forum. I would encourage you to go to aocs.org /Pulse Forum to get more information upon this meeting, which is being organized on your behalf to be held in Toronto this November. With that, I will conclude the formal part of my talk and be happy to take any questions at this time.
Yes, thank you very much, Dr. House, for this excellent presentation, very informative on defining quality of plant-based proteins. So it's the Q&A time. If you have any questions, please type in.
This is Amy with AOCS. It looks like I did get a question in my chat box, so I'll go ahead and jump in and read that one out while everyone else gets ready. The question is the genotype X environment data published? Can you share the reference?
Yes, not yet. We are just finalizing. So we have 1 year of data on the peas, and we have a limited data set on a variety of pulses. That limited data set is being ready for submission to publication. The pea is a much more comprehensive data set from a number of field sites within Manitoba. We only have 1 single year. We're collecting a second year on that in order to make a more fulsome data set. But we anticipate that we'll be collecting peas. We will be collecting peas from this harvest this year, provided we can sow them in the ground with all the water that's kicking around out there. But certainly we will be getting another year's harvest in order to fill out yet one more cropping year to add a year component to that analysis.
Does anyone else have any further questions for Dr. Howe? If so, you can write them in the chat box to the right. I'll give everyone just another moment. Okay, well, I think it was just Oh, wait, here's one. Is there a difference in extrusion and baking on digestibility values? Is it because of the matrix involved?
Great question. I think essentially what we're looking at with the difference in extrusion and baking, we actually don't understand the complete factors why we're seeing a suppressive effect on the digestibility values with baking. I am presuming that we are seeing some complexing of the proteins such that we're no longer having those proteins being released as they're binding with some of the reactive carbohydrates. It is something that we haven't looked at the specific mechanism why we see that suppressive effect with the baking, but it likely is a matrix effect as well.
Thank you.
Yeah, it is me, Janita, Dr. House, mentioned about nitrogen to protein conversion factor as 6.25 that is universally used for by the industry researchers and even in the trade. There's a very excellent review published by Journal of American Oil Chemists Society in 2019 January issue by Elaine Kroll, review with focus on soy protein. So that is, it looks at the pros and cons and evaluating different factors used by other um, non-soy proteins and especially the insect proteins and algal proteins that has come into the market. So I recommend that is a good read for those who are interested in the topic about conversion factors.
Yeah, thanks, Janita. Yes, it's an excellent review. And Elaine's paper was primarily focused on the soy question as to whether or not they should be modifying the nitrogen conversion factor of soy. Very exhaustive review, so it was a very well done publication. I encourage readers to look at that. I think the challenge that we have right now is in the official AOAC methods for measuring protein content. Right now, all proteins, with the exception of very limited numbers of ones that were cited within the AOAC, at least the AOAC approved methodology, require the use of 6.25, whereas some proteins such as almond, wheat, other tree nuts have different nitrogen conversion factors. And so we're talking, I see that there's a lot of interest in the protein space, and I see growing interest in this conversion factor. And I think it's a challenge to the research community to look at standardizing approaches to measure nitrogen conversion factor. I think we all can agree that 6.25 is not necessarily accurate, but it's heavily used. But we've had a lot of history of using that 6.25. So what are the implications of going away from that value to a more precise value that probably is more reflective of the actual protein or amino acid composition of that particular protein specifically? And so I think it is an issue, and I just see primarily because of competition, likely between, as an example, between the soy and the dairy sector, where there's pressure to look at capturing specific markets, that I would see that there's going to be growing interest in the use of nitrogen or challenging this value of 6.25. I do see a question here. Extrusion of pulse protein had higher digestibility. Is that because it's severe enough to create peptides, or to render them more available by disintegrating cell walls? I tend to think that it's the sheer force of that extrusion method that we're using likely is having a disruptive effect on the encapsulation of the plant proteins, would be my own position on why we're seeing the increased digestibility factors.
Any more questions for Dr. House?
me see. I think I may have gotten one more on my box. Nope. Okay, that was just a pop-up. Oh, yes. You said that TIA is decreased by thermal treatment so that the purest the protein-rich material, the less TIA content?
Oh, so certainly the trypsin inhibitor activity is decreased by thermal treatment, yes. And so certainly if you're dealing with an intact flower or an intact pulse, you're dealing with trypsin inhibitors. If you're isolating those proteins, fractionating the proteins, you are not only subjecting them to additional processing steps which can inhibit the trypsin inhibitor, but you're also isolating them away. So by purification, you're not only removing physically the presence of those inhibitors, but you're also suppressing them because there is some thermal addition to that process as well.
I think there is a follow-up question with the same line. So low processed material should contain higher amount of trypsin inhibitor activity or trypsin inhibitors?
Yeah, so if so the more the closer you are to the native pulse fraction in this case, the higher the trypsin inhibitor activity would be. So the more processing steps that you are adding, it's likely you're either diluting out the activity or through because they're heat labile, the addition of heat can actually cause a reduction in those trypsin inhibitors.
This is a new question. Are there any recent studies on absorption of bioactive peptides through enterocytes?
Certainly that's a question of bioaccessibility. I think it's a little beyond the area of research that my lab's involved in, but certainly there's a fair bit of work going on in bioactive peptides and the use of enterocyte models to look at absorption. I think there might be others on the line that are actually doing this particular methodology, but I would say the literature has examples of not only enterocytes, but other cell models and cell culture models that would be looking at peptide absorption. Peptides are very hot. I think there's a fair bit of work that's being done on looking at positioning plant peptides, and in particular from a nutraceutical or natural health product perspective.
There's a comment that thank you for this very interesting presentation from Frederick Bouvier. Another one following up. Can you tell us a few words about bioactive peptides found in pulse proteins?
Sure. It's not my area of expertise, but certainly a fair bit of work looking at the impact of hydrolysis on pulse proteins and the release of particular peptide fractions. That I know there's work going on even in our own institution looking at the antihypertensive effects of yellow pea hydrolysates. And so there's a fair bit of work going on looking at hydrolysis, the purification of those hydrolysates, and then the assessment of their overall bioefficacy and bioactivity. And so I think that's one of the fascinating things about plant-based materials. They provide a wealth of variety and wealth of opportunities for focusing on the use of either fermentation or enzymatic hydrolysis to look at novel peptide and bioactive fractions that can be created.
And this is Amy again. I got a message, I believe, sent to me. Excuse me. Year-to-year anti-nutritional factor variability in pulses, is that an area of interest of your lab? And can you give a general comment about this topic?
It certainly is an area of interest. We're just initiating that as part of our screening protocol right now to look at not only trypsin inhibitor activity, but also reactive lysine, particularly for those where we've done a processing step. We have not published in this area. Others have published certain elements of anti-nutritive factors, but I think understanding variability is really at its initial stages. And so I think when we're talking about the breeding programs for most of the major crops, the primary focus is on agronomic quality and yield traits. I think yield and disease resistance are two of the key factors that are going to continue to drive interest in and scope the breeding programs moving forward. But there has been increasing interest and awareness of the tremendous opportunities that can exist, in particular when you start positioning identity preserved production systems that can select a plant source as a high in a particular fraction that would benefit a particular industry segment. For example, a yellow pea that happens to be high protein yellow pea that might be better suited for wet fractionation methods, for example. So I think though there's tremendous interest in looking at how we can add more value into the breeding programs by focusing on other quality parameters such as nutritional quality and functionality. And so certainly that's been an emphasis in the work that I've been doing and also in the labs of the collaborators that I work with both here and at the University of Saskatchewan, including Mike Nickerson and Janita as well. So.
Awesome. Well, Janita, did you see or have any more questions? I'm not seeing any.
No, I don't think so.
Well, thank you everyone, to Dr. House as well as Janita for bringing us this information, and thank you everyone for joining us. I really appreciate it. It's very exciting.
Great, thank you very much, and I appreciate the opportunity to share our findings and share some thoughts, and I thank you all for your attention. Have a great day.
Thank you, all participants and AOCS and the presenter, Dr. House and Amy, also connecting all of us.